classes, we have discussed about the general uh, morphological characteristic and the general life cycle of a trematode, of a cystode, of some nematode. Okay, then we will continue here in today's class is the general life cycle of certain nematodes. If you must remember, in the nematode, that means the parasites under nematode means we are particularly interested in one class, that is nematoda. Under this one, we are having, as I have said in the last class, that we are having two subclass. One is Caesarnensia and another one is Adenophoria. Okay, so earlier the classification of a nematode is based on phasmida and aphasmida. Okay, what is phasmid? Phasmid and aphasmid, I mean, it is derived. The name is derived from. There is one in case of nematode, as I have told you that they parasites. They are a bit higher organism as compared to trematode and cystode, tapeworm and flukes. So they are having a, in addition to reproductive organ and in addition to their alimentary canal, they are also having a tactile organ. That means the nervous system. Okay, so I'll tell you what are the nervous system and all this. But for our classification here today is that there is a nervous system or a tactile organ at the anterior portion, tactile organ, okay, at the anterior portion known as amphids, A-M-P-H-I-D-S, amphids. And posteriorly there is a tactile organ known as phasmid. Okay, so based on the presence of whether the parasite is having phasmids, that means the posterior nervous organ or tactile organ or not. Parasites are many times classified as phasmida and aphasmida. Most of the parasites of veterinary and human medical importance belongs to phasmida. That means which are having posterior nervous post nervous organ. Okay, the tactile organ. So here in today's class also, we are continuing with the subclass Caesarnensia, or earlier they were known as Phasmida. So under this one we are having four order. One is Strongyelida. We have, let's look back. Uh, let's see the pointer. We are having uh, Ascaridida, four order. Another is Strongyelida, Rhabditida, and Spirulida. Okay, Ascaridida general characteristic and some general life cycle we have talked about in the previous class, that parasite is laying oviparous egg. Okay, that means the larva does not contain the egg does not contain a larva, and the infective stage is the egg containing the larva, which is encapsulated within the eggshell. As the eggshell is very thick and the larva is incapable of hatching out in the environment. But of course, they will hatch out in the intestinal environment, in the intestinal lumen, with the presence of certain bile salts, acids, hydrochloric acids, and pepsins, and taurocolic acids, and anaerobic condition. And all this we will talk about later on. But the parasites tick larva, the egg is unable to hatch in the environment. Okay, this is the general life cycle what we have discussed in the last class. In today's class, we will talk about the parasites under the order Rhabditida. Okay, so one thing which should strike your mind in Rhabditida is that the parasite lay ovoviviparous egg. That means the larva which is contained within the eggshell. At the time of laying of egg, the egg shell contain a larva. Okay, but this eggshell is very thin, unlike the big thick fat one of ascarid. So they are readily hatched out in the environment or even in the intestinal lumen itself when the egg suppose the egg is present, the parasite is present in the small intestine. During its passage through the intestine, the egg will hatch out into the larva many a times. Some egg doesn't hatch, but some hatch out. Okay, in the environment, it will develop, unlike ascarid worm, it will develop into L1 and L2 and L3, which is which is known as filariform esophagus. I told you in the previous class that the parasitic larvae or parasitic larva singular can be differentiated morphologically also. If you see the esophagus, okay, I'm going back to there. Okay, if you see the esophagus here, okay, this is there is one bulb here. Okay, there is another bulb here. That means there is two bulbs. That means the parasite is rhabditiform. I told you that RHAB, rhabditiform. Okay, so if the parasite does not have a bulb, if it is just a plain structure, that is known as filariform. Filaria means hair-like or thin. Hair-like or thin. That means the most of the bulb are absent. Why it is so is because the larva 1 and larva 2, those are the larva 1 and the larva 2, or L1 or L2, they are actually, they are free living in the environment. That means they are feeding, they are eating something. They are eating decaying plants, decaying material. Some even eat mold, some even eat fungus, some even eat bacteria. So for that nutrition, they require muscular esophagus. But when the larva turn into L3, that means when it develop into infective stage, the larva does not feed anymore. The larva depends on the food storage of L1 and L2. Okay, so when the larva depend on the food storage of L1 and L2, the esophagus is modified in such a way that it no longer required muscular structure. So it is just a plain capillary tube-like structure. In fact, L3 never fits. It is covered with the sheet of L2 and L3 itself. So there is no mouth opening at all. So that is why the larva of filariform or the larva of L3 is known as, I mean, it devoid of the muscular structure, so it is known as filariform. The other two, the previous L1 and L2, they are feeding, that's why they are known as rhabditiform esophagus. Okay, so the esophagus is, the infection is continued by many a times that they can penetrate, these larvae can penetrate the skin, or sometimes the infection is continued in the definitive host, I mean to say. Okay, so the L3 which is present in the environment will give infection to a host, that means to the mammalian or to human being, okay, by ingestion or by skin penetration. Here the the life cycle is a bit unique here. Please listen to me carefully, follow me carefully, this is important. Okay, so the adult male and adult female, they reside in the small intestine. In case of Rhabditida, they laid eggs. As I told you that this is the egg, which is, you see, very thin eggshell, which is containing a larva. The larva, which is packed within a very thin eggshell, okay, which is stuffed within a thin eggshell, is readily hatched out in the environment. That's why this larva is containing the larva is containing a L1 L1 type of larva. Okay, the larva is an L1 type. That means it is containing the double bulb esophagus, BULB. Okay, so in the environment, when it comes to the environment, they can develop into adult free living or adult free living male, free living adult male or free living adult female. Why it is so? Remember at the beginning of the class, I mean at the beginning of the course, I told you that the intention of the parasite is not always to cause a disease. The intention is rather to live and let live, to reproduce and to populate in the environment. Okay, so if the environmental temperature is favorable, that means if there is a warm, sufficient warm, 25 to 30 degrees centigrade, even up to 35 degrees centigrade, or and there is sufficient humidity, they will not develop. They will develop into adult male and adult female. That means this life cycle is known as heterogonic life cycle. HETERO, heterogonic life cycle. Okay, I'm repeating. If the environmental temperature is favorable for the parasite survival in the environment, they will develop into adult male or adult female. But if the environmental temperature is not favorable for the parasite, that means it is very harsh environment. It's very harsh environment. It's very warm, or it's very sultry and dry and very arid condition, or if it is very there is no moisture at all, as I told you that it's very arid condition, that means a desertic condition. Then what will be there? They, instead of developing into L1, I mean adult free living or adult free living, they will directly develop into the L3. That means they will subsequently develop into an infective stage. This is known as homogonic life cycle. Okay, subsequently, if the adult free living and adult free living male and adult free living female also, they will copulate and will give birth subsequently to a, later on they can give to a reptitive, I mean the filiary form esophagus type of larva. Okay, so what does it signifies here is that what do we learn from here is that the intention of the parasite is not always to cause a disease. The intention is to live along with the host in harmony with it. But if the environmental temperature is such a harsh condition, so they said yes, why to risk ourselves in this harsh environmental condition? Let's enter into the host. That's why they rapidly develop the adult, I mean they would rapidly develop into the infective stage, and subsequently what will be the next stage of the infective stage? They will penetrate the skin and they will continue their life cycle in the host. If the environmental temperature is favorable, they will continue their life cycle in the environment. If not, they will continue their life cycle in the host, because the host will provide some better environment as compared to the external environmental conditions. I hope I'm clear. So heterogonic life cycle, homogonic life cycle, we many times ask this one in exam. Okay, so the next one is the parasites under Strongylida. Most of the uh, tropical animal health, uh, tropical animal production is hampered by this group of parasites. Okay, most of our focus also will be on these four superfamilies: Strongyloidea, Trichostrongyloidea, Metastrongyloidea, and Metastrongylo ankylostomatoidea. If we go very much in deep, there would be thousands of the parasites under Strongylida order. Their number is huge, but we will try to cut short and we will give some important points in the Strongyloidea. The parasites mostly belongs to horse, strongyle. Okay, the next one is Trichostrongyloidea, mostly ruminant strongyle, ankylostomatoidea. This belongs to mostly hookworms. Metastrongyloidea, mostly lungworms. Okay, so the parasite lays this type of egg. Of course, this is oviparous egg, but unlike ascarid worm, it is having a thick, a thin eggshell, and there is segmentation of embryo. I'm just going back so that it will be clear to you. Okay, in case of ascarid worm, there is no embryo. There, of course, there is an embryo, but there is no segmentation here, and the eggshell also is very thick here. But here, in this case of strongyle, we generally call it a strongyle also. You can call it a strongyle. Okay, the strongyle, the egg contain the segmented embryo with a thin eggshell here. Okay, so why do we call it a strongyle is, I'm just going back here in the picture. Okay, this is a strongyle adult male. Okay, so let's recap what we have discussed at the beginning of the class. The parasites, they are mm, they are round in cross section. Point number one. Point number two, they are tapering at both the ends. In case of fluke, they are dorsoventrally flattened. In case of tapeworm, they are dorsoventrally flattened. Okay, but here in this case of nematode, they are round in cross section, just like a pen or just like a pencil, but they are tapering at one minute. So the parasite, they are tapering at body ends. Uh, but in case of a male strongyle, there is a, where's my pointer? Okay, there is a posterior, what should I say? Enlargement at the posterior end. There is enlargement at the posterior end. This is known as bursa. Okay, the function of this bursa is it will embrace the female during copulation. Okay, so this bursa is supported by bursal rays. These are the bursal rays. Okay, these are the bursal rays. But this is the spicule. This is the, I told you that this is the gubernaculum. These are the pair of spicule. S-P-I-C-U-L-E. Okay. So here, if you see the general life cycle, the adult female will lay oviparous egg with segmented embryo, segmentation of the embryo, and it will develop into filariform type of esophagus in the environment. Very simple life cycle. The egg will be laid and it will come out in the environment. It will hatch if favorable environmental condition is present. If there is favorable environmental conditions, within a week it will hatch into L1. L1 will develop. L1 is having the rhabditiform esophagus. It will feed. Subsequently it will convert to L2. L2 will continue to feed. Subsequently, it will develop into L3, which does not feed anymore, and L3 will depend on the food storage of the parasites and food storage inside the inside its body. Then subsequently it will wait for a chance to infect into the definitive host, into the host. Okay, one thing what you must also remember here is the adult parasites, they live or they survive with the food storage. The food storage is in the form of lipids. Please mark it. Food storage of adult parasite is based on lipid, but whereas in case of a larva, it is glycogen. The food storage is in the form of glycogen in case of larva, but whereas in case of adult parasite, it is lipids. Okay, so infection of the host occurs sometimes by penetration of the skin by the infective stage, that means by the infective filariform esophagus in case of ankylostom, that means hookworm, or sometimes they also would be ingested through Parparos infection. Okay, sometimes the parasites under Metastrongyloidea superfamily, they are having an indirect life cycle. They are having an indirect life cycle. That means they would require the intermediate host, earthworm. Okay, sometimes snails, sometimes slugs. Okay, land snail and slugs, they require the intermediate host. Okay, so but in this case also, in this case of Metastrongyloidea, even if they require an intermediate host, the infective stage is still remains as L3. That means the L1 will enter in the intermediate host, it will develop into L2, and subsequently it will develop into L3, and this L3 will be ingested by the definitive host. The details we'll talk about later when it comes to the systemic part. Okay, so this is a life cycle, the pictorial diagram. The adult parasites of Haemonchus contractus disease, and that it is one of the most important, not only on parasites, but it is one of the most important infectious disease in of small ruminant in India. I'm repeating, it is one of the most important infectious disease of small ruminants in India. The morbidity is 100%. Okay, if you cut open sheep or goat abomination, not much in Northeast, but in other parts of the country, every small ruminant would contain these parasites. They are the one which is causing anemic conditions. They draw lots of blood, and they of course they do not cause mortality, but due to their high morbidity, then due to anemic condition they cause, there is a huge production loss due to Haemonchus contortus. The other parasites reside in the abomasum of the host, that means sheep and goat, and the eggs will be passed out in the environment. In the environment, it will develop within a week into L1, L2, L3. Okay, so L3 will wait for the host to ingest. Okay, they would climb up to the grass blade, and subsequently during grazing, the definitive host would ingest and completes the life cycle. It's very simple life cycle, but the the more it is, means the simpler the life cycle it is, and the simpler the parasite chance of completion of their life cycle. If it is very complicated, they do not want that complication also. If that is complicated, and it will be difficult for them to complete their life cycle. Okay, that's why they want to develop their life cycle in such a way that it is more simplified, and when it is more simplified, the chance of survival is more. All right, I told you that they are very clever. We will come to see when we study the systemic part, and you will come to realize that uh, there are many interesting things and many intriguing things that you will find out, and it is very interesting to study parasitology, despite your seniors might have told you that it's very dry subject. Of course it is very dry subject, but it is very easy to forget the thing. Okay, and once you, as I told you at the beginning, if you turn one page, it is very simple to learn, and the next page you turn, everything goes volatile. Okay, so anyway, if you go through whatever I am telling you in the class, and things will not be difficult at all. It's very simple. Okay, the next one is Spirorida order. These Spirorida, they are unique. Okay, you might have heard, you might have seen also, especially those from North India and some parts of from the mainland India, that elephantiasis, and others also you might have seen in textbook or in the television, or maybe in the from the internet, you might have seen that elephantiasis. This is one of the most important disease of human being in India. In India, there are, they say, National Vector Control Board, mm, of the Ministry of Health. They say there are 6 important infectious disease. One is malaria, one is that is a protozoa, another one is kala-azar, that means leishmaniasis, another one is what's the elephantiasis. Okay, the next one will be dengue, JE, and TB. Okay, so others are, and don't say they are not important, but these are the prime important disease of Indian in human health. Okay, so here spiroid order, they are mostly I mean, of course, there are filarial worms are very important, but apart from that one also, they are certain parasites which are important in animal health. Okay, here the parasite, the characteristic is that they are not laying eggs, but rather they are laying down, they're giving birth to a larva. Okay, the L1 is found in the many times the parasites, they are in the blood vascular system. So the larva L1 are freely, you can see, freely swimming in the blood vascular system. Okay, so usually they are having heterogeneous life cycle. That means they are having an intermediate host. That means they are requiring, I mean, they are having an indirect life cycle. Okay, so the L1, which is freely swimming in the environment, are ingested by the intermediate host, mosquitoes. Okay, so when mosquito feeds on the another host or the same host, the inside the mosquitoes, after it develop into L1, L2, L3, and it inject the new into a new host. It inject into a new host or the same host. Okay, so this is the life cycle of a spiroid. As I told you that the other parasites, they are sometimes in the heart of, here in this case it is the Dirophyllary immitis. This is the heartworm of dog. Okay, so the parasites, they are mostly located in the pulmonary artery and the right ventricles. Okay, so when the parasites locate in the right ventricles or the pulmonary heart, so the larva is, you can see, in in the blood, okay, in the plasma, they are swimming. So when mosquito comes and bite, they ingest the L1. Okay, inside the mosquito, they will develop into L1 or L2, and subsequently it will develop into L3. So when mosquito bites the another host or the same host, then subsequently the they will inject the L infective stage, and the cycle completes. So this is the life cycle of a spiroid. Okay, this is a general life cycle, and there, of course, there are some uniqueness and some differentiation that we can see in the systemic part. Okay, so for the last one is that the parasites under subclass. Now I'm jumping on another subclass. Earlier, whatever we have discussed was uh, uh, under the Sessionensia or Phasmida. Here it is Adenophoria or a Phasmida. Okay, here in this case we are going to see only one order, that is Anoplida order. Okay, under this one we are having three uh, family: Trichuridae, Trichinellidae, and Capillaridae. Trichuridae and Capillaridae, they are very simple, just like your strong guide. The egg will the egg will be laid in the environment to develop into L1, L2, L3 ingested. Simple as that. But Trichinellidae, they are having an unique life cycle that I'm going to explain to you just now. Okay, so the Trichinella, we know that the parasite is a permanent parasite. That means they do not require the environment, they do not require to come out in the environment for completion of their life cycle. They would remain, they would be transferred from one host to another host through predations. Okay, so let's see. In case of Trichinella, here it is given swine, the definitive host is pig and other carnivores. Let me give an example here. Okay, let's say this is this is mouse. This part is mouse. Okay, let's say this part is mouse. So thing is, inside the mouse, after the Here it is man is given, but I'm giving example mouse or any rodent. Okay, here in this case, when the rodent ingested the larva, when the rodent ingested the larva, it will develop very rapidly. Within 4 days, they will develop into adult male and adult female. They will develop into adult male and adult female, and they will copulate for some time, and the adult male will die, and adult female will live for 2-3 days more, and she will lay a larva. This larva will be laid in the intestinal wall. Okay, when the larva laid in the intestinal wall, what will happen? The the larva will access to they will access to lymphatic channels, and they will be migrated throughout the body. They will be taken throughout the body, and inside the muscular tissue, they will develop and they will encase or they will coil themselves. Okay, this is the larva. This is the larva which is coiling up inside the muscular tissue. This is known as nurse cell, N-U-R-S-E. Okay, what does the nurse cell do? Nurse cell provide nutrition, and they will also taken out the excrement of the larva. They have drawn out the excrement, and they also supply for nutrition. Okay, so this nerve cell, the larva within the nerve cell is waiting for the another host. That means what this is a mouse. Let's say this is another one is cat. It is waiting for cat to ingest this mouse. Okay, so what will happen inside the cat once cat have feed on the that rodent? What will happen inside this cat? Inside the cat, they will develop into adult male or adult female again. Okay, so inside this cat, they will develop into adult male and adult female, and the larva will be laid by the female again, and this larva will be subsequently it will be taken to various tissues and it will encase again. Okay, so what do I mean to say here? Please follow me carefully. When the time when the parasite develop adult male and female, at that time the parasite is known as, and means the host is known as definitive host, and the time when the host contain a intermediate host, or here also the time when the host contain an intermediate stage. That means the larva at this time it is known as intermediate host. The time when the parasite, I mean the host contain the adult male and adult female, at that time it is known as definitive host. So what is the meaning here? There is auto-heterogeneous life cycle. At the beginning of this class, at the beginning of the course, I told you that when it is the life cycle, at some point of time, the host act as a definitive host, and subsequently it act as a definitive host for cat. Okay, so this type of life cycle is known as auto-heterogeneous life cycle. Is it clear? I'm just repeating. Let's say there is one rodent which ingests the larva of the larva of the larva of Trichinella spiralis through garbage. Or let's say there is one wild boar that is lying there, dying over there, and the rodent feeds on it. Okay, so during the feeding, they would ingest the larva of Trichinella spiralis. Okay, so after 4 days, they will develop inside the intestine. They will develop into adult male and female. At this time, the host is known as a definitive host, but subsequently the host, I mean the adult parasite very easily die within 3-4 days, and within 5-6 days, within a week, the adult parasites vanish, they died. Okay, so what will happen? The larva would be laid down by the adult parasite, then the larva will insist in the muscular tissue or inside the visceral organs. So what will happen? So so at this time, it is acting as an intermediate host for the next host to ingest. Okay, so this type of life cycle is known as auto-heterogeneous life cycle. I think I'm clear. Okay, with this one we complete our general, the general life cycle. If anyone is having any doubt, please let me know. You can ask. You can just type on the chat section, or you can unmute yourself